Thank you. Good afternoon. My name is Amanda Lawrence. I am the Fellowships Coordinator with the National Sea Grant Office, and I'll be introducing our speaker, Samara Nehemiah today. But first, I just want to thank you all for joining. This is our first speaker of our seminar series, and I want to thank the NOAA Library for hosting. So we can get started. Samara Nehemiah is a PhD student at the Chesapeake Biological Laboratory, working on developing new statistical models to estimate abundance of fishes in the Chesapeake Bay. She's a current NOAA NMFS-Sea Grant Population and Ecosystem Dynamics Fellow, working with her mentor, Dr. Amy Schuler from the Southeast Fisheries Science Center. And today, she will be presenting her talk, Estimating Striped Bass Abundance in the Chesapeake Bay using spatially explicit population models. So take it away, Samara, when you're ready.
Awesome. Thanks so much, Amanda, and thanks everybody for tuning in today. Today I'll be talking a little bit about my PhD work, which is to develop spatially explicit population models for striped bass in the bay. So some background, in the United States, fisheries management can typically be thought of as the cyclical process that you see here on the right, where there is some method of data collection, which is used to develop quantitative models called stock assessments that help determine stock status. Then a series of management recommendations are proposed based on that assessment, and public input is sought out, and then management can determine if regulations should change or not, and then this cycle kind of continues over and over again. And stock assessments are a crucial part of this management process in order to understand the current status of a fishery. These models estimate abundance, recruitment, mortality, and other metrics, and are also used to determine if a stock is being overfished or undergoing overfishing. But there often exists some inconsistencies between stock assessments and the management, where spatial scale that stock assessments typically model might not match the spatial scale of management for the same species. Stock assessments often make assumptions that help to simplify the complexity of assessment models. So for example, many stock assessments model a population that is assumed to be homogeneously distributed and well mixed across its range. But we know that most fish populations experience some sort of spatially varying natural mortality, fishing mortality, abundance, or other population dynamics. So modeling a homogeneous population violates the naturally occurring dynamics of a stock and can create bias in model estimates and provide incorrect reference points for management purposes. So developing spatial population models can allow for management scales to match their assessment scales, but also incorporating spatial structure into stock assessments adds some biological realism into the model for species that don't exist homogenously across space. So the use of spatial stock assessments has been limited in the past because it often requires a lot of additional data across multiple spatial regions. For migratory species, it might require some knowledge of movement or tagging information that isn't always available for species of interest. And these models are really complicated and they take a lot of time to adjust and fine tune. And management typically requires that stock assessments be completed within a few years or on short timelines. And so there's usually little time that can be devoted to developing new methods or models. And so incorporating these models into management can actually be really challenging. But recently, the use of spatial models has increased as a result of additional computational abilities and computer softwares. And although many studies have shown that increasing spatial complexity to match sort of the biological characteristics of a population can actually improve model estimates, there are still limited spatial stock assessments that are used in practice. And so kind of just moving on to my study region. So the Chesapeake Bay is in the mid-Atlantic of the United States and is important habitat and spawning grounds for many fish species. And although the Chesapeake Bay supports many important commercial and recreational fishes, there's actually little information about how populations of fishes have changed in the bay itself. Excuse me. Spatial population estimates are particularly beneficial for the Chesapeake Bay region because this region kind of has its own set of challenges as a result of climate change, its land use within its watershed, and also experiences various fishing pressure. And many species within the bay have a life history strategy that includes some sort of migration into or out of the bay at different life stages. And so when available, spatially explicit models can help forecast how future environmental conditions and management decisions may actually affect fish populations and ecosystem dynamics. And so the overall goal of this project will be to eventually get bay-specific population estimates for numerous fish species of interest, but striped bass were selected as a pilot species for this project to develop new methods for spatial models in the Bay region. Striped bass support an important recreational and commercial fishery in the Bay and along the Atlantic coast. And many states along the Atlantic coast closely monitor and regulate striped bass in state waters, and therefore there's a lot of data that's available for this species in order to develop new statistical methods. And they're also highly migratory and exhibit natal homing strategies. So that means they return to their spawning areas in the rivers every winter to spawn. And so just kind of moving into some of the management for Atlantic striped bass. So Atlantic striped bass are managed by the Atlantic States Marine Fisheries Commission, or ASMFC, and they're currently modeled as a homogeneous population from Maine to North Carolina, but management is implemented at the state level. And so within this kind of assessment unit, they're considered to have 3 major spawning areas that's indicated here on the map to the right, the first being the Chesapeake Bay stock, the Delaware Bay stock, and the Hudson River stock. But they also have smaller spawning areas along the coast and another kind of major spawning area that's not considered part of this assessment unit. But the Chesapeake Bay is definitely the most productive spawning area for this population, and studies show that at least 60% of the Atlantic coastal fishery comes from the Chesapeake Bay stock every year. In the late 1980s, this population was considered to have collapsed, and a moratorium was placed on the Chesapeake Bay fishery. That fishery recovered and increased over time, but the most recent update from 2022 indicated that striped bass are not undergoing overfishing, but are currently overfished, and so they've been working on recovery plans to recover the stock within a few years. And so this species exhibits great spatial complexity, and by disregarding spatial structure in those stock assessments for the species, we really could reduce how effectively we're managing this species. So that kind of brings me to kind of the overall objectives of my specific research for my PhD. The first is to develop a statistical catch at age model to estimate abundance of striped bass in the Chesapeake Bay. The second objective is to assess the performance of that spatially explicit model and determine kind of how the performance of the model changes as we have different data availability, and kind of determine what the data needs are for a model of this kind. And the third objective is to kind of determine how the performance of this model changes as a result of climate change. So we expect this species to its distribution or productivity to change as a result of climate change, and we kind of want to see how our model might be able to track those changes. But for today's talk, I'll just be focusing on this first objective, going over the model structure, what goes into it, and kind of where we are today. So talking about kind of the model overall and kind of how it works. So our model tracks ages 1 through 15, where the 15 is a plus group. This model also considers 2 biological stocks for this population, which are shown in the circles on the graph to the right. So the first is the Chesapeake Bay stock, and the second is what we're calling just an Atlantic coast stock, which includes the Delaware Bay and Hudson River populations. We also have 2 spatial regions to kind of match that biological structure, the first being the Chesapeake Bay, which is shown in this red box, and the rest also being called the Atlantic coast, which is kind of in coastal waters, everything else outside of the bay from Maine through North Carolina, coastal North Carolina. Our model runs from 1985 to 2017 and has 2 subannual time steps that kind of account for striped bass migratory behavior. The first is January through June, which is when we assume striped bass are spawning. And then the second time step is July through December, when we assume striped bass are in their coastal regions migrating. And this model will be able to estimate abundance for each stock in the 2 spatial regions, recruitment for each stock, fishing mortalities will be estimated for the 2 regions. We'll have estimates of biomass for each stock and region, and then we'll also estimate a movement parameter. And so just to kind of quickly go over how our model is set up, I first want to explain how we're calculating abundance in each region and how we're keeping track of that. So rather than kind of keeping track of migration rate, we're actually considering what we're calling an occupancy probability. And the occupancy probabilities can be defined as the probability at age that a fish from a certain biological stock is found in either the region or the Chesapeake Bay spatial region. And so just kind of mathematically what this looks like in these equations. In our model, we're keeping track of both the total stock abundance for the combined spatial regions, which we're calling NT here, and then we're also keeping track of abundance for each stock in the specific spatial regions, which we're calling N. And so to kind of go over how we're, fitting abundance in these equations, to get abundance, we first need to calculate the total stock abundance, and so that can be found as the number of fish from a specific stock from the Chesapeake Bay spatial region that survived from the previous time step, and you can add that to the number of fish from the same stock in the Atlantic Coast spatial region that survived from the previous time step. And then once we have our NT, we can then kind of distribute that overall stock abundance into its specific spatial regions by applying this occupancy probability here. And so that just kind of helps us, it's easier to estimate than keeping track of migration rate for this model. And so just with that in mind, we can now kind of visualize how this model works kind of quite simply. So just looking at this graph to the right, we have fish from the Chesapeake Bay stock in this reddish kind of brown color. Fish from the Atlantic Coast stock are in this blue color. And so for our first time step, where in the beginning of the year, we assume striped bass are spawning. And so you'll see mostly bay fish in the Chesapeake Bay spatial region and Atlantic Coast fish in the Atlantic Coast spatial region. But then in the second time step, we assume that striped bass are migrating. And so you'll see fish leave the Chesapeake Bay, enter the Atlantic Coast spatial region, and then we also assume that there are some level of resident fish that don't migrate throughout the year, and these are primarily made up of younger striped bass. And then in the next year, we're back to our first time step when striped bass are spawning. So you see those striped bass leave the Atlantic Coast spatial region and enter the Chesapeake Bay, and this cycle kind of repeats. So that was kind of how our model works. Now moving into kind of where our model is right now, and talking about some of our data inputs, starting with our fisheries independent data. So this model utilizes 7 adult surveys that are indicated by the circles here on the right graph. 2 of those surveys are in the Chesapeake Bay. 5 of the adult surveys, are in the Atlantic Coast region, including 2 spawning stock surveys with 1 in either spatial region. And we also have 2 H1 surveys in this model that are indicated by the triangles here on the right, and with 1 in either spatial region. We have 3 young of the year surveys shown in the squares, where each of them kind of exists in those major biological kind of stock areas. And I will point out that for the most part, most of our surveys either sample in either the first or second time step, but there are a few that sample throughout the year, and so they split the time steps. And for those, we kind of disaggregated the data, and so we have indices of abundance and proportions at age for each of these surveys and their respective time steps. We also have occupancy probabilities as data inputs for this model. So these are from the output of a mark recapture model that utilized tagging data that was developed by some of our collaborators at the Virginia Institute of Marine Science, Adina Schoenfeld and Rob Latour. And so these are input and used as informative priors for our probability estimates in our model. And then we also consider an aging error matrix to account for how we're getting aging data from our surveys, and I'll touch on that in a second. And just a note on some of our data inputs. Most of our inputs are actually not stock specific. They kind of apply to a region, with the exception being the occupancy probabilities are stock specific. So just to keep that in mind of kind of what our data looks like as we're walking through the model a bit. And so quickly, just to kind of go over what we mean when we're talking about aging error. So many state agencies use scales to age striped bass rather than otoliths, but otoliths are typically a lot more reliable than scales for this species. And looking at this plot that was made by my former lab mate, Sam Shiano, she kind of took all of our data sources that, that have ages from both scales and otoliths for the same fish and plotted them on this graph. And what you'll notice, particularly for the ages 9 and above, that scale ages commonly and sometimes by quite a bit underestimate the true age of a fish if we consider the otolith age to be its true age. And so we kind of want to account for this in our model. And so we have an aging error matrix for ages 15 plus that we apply to all of our surveys that utilize scale ages. Moving on to the fisheries dependent data inputs, we have kind of one fleet for each spatial region and time step. That includes a total catch and the proportions at age. So for each spatial region and time step, it includes recreational, commercial fisheries combined. And so just kind of what that looks like, it's made up of the recreational catch that is retained, and we get that data from MRIP. It also includes the commercial catch that's retained, which we get from the state agencies. And then it also includes some proportion of both of those fisheries that are discarded that we assume have died, and that is the percentage of that is dependent on the fishery. Moving into some of more of how our model is structured, some of the assumptions and how we start the model. So we assume that we have a natural mortality at age that is constant over time. We also estimate abundance in the first year in equilibrium. Recruitment in every year is estimated as independent parameters for each stock. So we have an average recruitment that deviates for each stock. And then catchability, we have that varies for every survey. And again, for the surveys that split the time steps, we have a different catchability for each of those time steps. Moving into how we're estimating and modeling selectivity. So we utilize, in this working version of the model, we either have a logistic or a double logistic function. So we use the logistic function for the Atlantic Coast fishery and the 2 spawning stock surveys. And then we utilize a double logistic for the other fishery and the remaining surveys. And for the survey selectivity, we have constant selectivity over time, but again, for the surveys that are across the time step, we have different selectivities in both of those time steps. But then for the fishery selectivity, we vary through time utilizing 3 selectivity time blocks. So the first, to account for like changes in regulation over time. And so the first time block we have is a moratorium time block from 1985 to 1989. We also have a post-moratorium time block from 1990 to 2014, and then we have what we're calling just a recent block from 2015 on. And then finally, just some information on kind of how we're fitting the model, our likelihoods and penalties to keep our model in check. I'm not going to go over all of these in too much detail, but one thing I will point out is this binomial likelihood that we have for the proportion of each stock in the coastal fishery. And so I mentioned that studies suggest that at least 60% of the coastal fishery is comprised of the Chesapeake Bay stock. And so we kind of use some of the recent literature that was conducted in our model time period to kind of utilize as in a likelihood to kind of balance out the how we're estimating abundance and how the model is fitting abundance. Without this likelihood, the model tends to want to kind of kill off one of the stocks and kind of fit the model only using either the bay or the coast stock. And so by adding this likelihood, it kind of helps keep the estimates more realistic and more on balance with each other. And so the studies we're using here are Hasegawa et al. 2022 and Kneebone et al. 2014. And so that was a lot on kind of how our model structures the data we're using for it. And now I'll kind of show you some of our results. I will say that we're still kind of developing the model, and so these results aren't final and they're likely to change within the next few weeks, but still exciting to look at nonetheless. So on this side, you can kind of see how our model is estimating abundance. And so kind of how these plots are set up. The names here at the top of the graphs indicate which spatial region they use abundances are for. The numbers represent the time step in our model. We have year on the x-axis, population on the y-axis, and the colors indicate which stock these fish are from. And so something you can notice in all of these time steps is that the model is able to estimate low abundance in the late 1980s when there was a moratorium. That population then increased over time. And then recently we've had some declines, but it has fluctuated a little bit. And I will point out that these abundance plots are for the total population for all ages. So they're really heavily influenced by recruitment or age one, some of the younger fish. And if you actually plot out the adult fishes or age 7 and above, you'll start to see in the Atlantic coast region, the Chesapeake Bay stock will be much higher than the Atlantic coast stock, which is what we kind of expect and what our studies suggest. Some more results, we can also look at how our model is kind of estimating the parameters and our selectivity function. So this is for the fisheries selectivity. The first row here indicates our moratorium time block from 1985 to 1989. The second row is the post-moratorium time block from 1998 to 2014. And then the last row is our recent time block. Again, these are for the fleets or which spatial area, the names here, and then each time step. And so what you'll see is, in general, this model is having a tough time kind of sometimes getting reliable estimates of selectivity, particularly for the moratorium phase. This could be due just to low sample sizes from that time period, but something to keep in mind, we might have to put some stronger penalties or change our bounds for these functions. Moving on to some of our fisheries independent data, we can also look at how well our model is estimating the indices of abundance for some of our surveys here. So these plots here, the names at the top correspond to the survey or the state that the survey is conducted in. The blue line is the observed indice of abundance, or what we have input into the model. And the red dotted lines are how the model is estimating that abundance over time. And so looking at these different surveys, you'll see that for the most part, our model does a decent job at fitting through the data. It kind of goes through the middle of these data points, but we do have some exceptions. I'll point out here just the Maryland spawning stock survey. This model has a kind of tough time fitting the trends of this survey, and that's just because the trends kind of differ from what some of the other surveys say. So it's just something to keep in mind as we work through our results and consider our data inputs going forward. We can also look at how survey selectivity kind of fits for the surveys. Again, the names correspond to the survey or the state that the survey is conducted in. Again, most of these actually make pretty good sense for our surveys, but the model does kind of estimate some steep slopes. And so again, playing around with maybe some of those bounds or penalties functions for these relationships. And then finally, the last kind of result, a plot I'll show are the occupancy probabilities that our model is estimating. So these plots are a little bit different. The names on the top of these plots indicate which biological stock that these probabilities are for. The numbers are the time steps, and what these plots are showing are the probability that a fish from this stock is found in the Atlantic coast spatial region. And so if you take 1 minus any of these probabilities, you'll get the probability that that fish is found in the Chesapeake Bay spatial region. And here the blue lines are the priors, so the outputs from the marker capture model that we enter into the model, and then the red lines are how our model is estimating occupancy probability. And so just to kind of point out, this kind of shows kind of movement, our movement of striped bass into the 2 spatial regions. In general, not many fish from the Atlantic coast stock enter the Chesapeake Bay spatial region. You'll notice that most of these probabilities for the Atlantic coast are hovering around 1. And then just to kind of get out how our, most of our movement in the model works, you'll see that Chesapeake Bay fish, particularly in the second time step, increasingly are found in the Atlantic coast in the second time step with age. So that makes sense. That's kind of what we expect is happening and what our studies suggest. So that just kind of gets at movement for this model. All right, so that was just kind of a summary of kind of where our model is, how it works, but we still have a few adjustments to make to our model. So first, we're hoping to extend out the terminal year of this model with updated data. We're hoping to model through 2019. I also pointed out that selectivity can sometimes be hard for this model to estimate. I pointed out that we're either in this model, either using a logistic with 2 estimated parameters or a double logistic with 4 estimated parameters. But when we start to introduce stronger penalties on those parameters or change the bounds, we actually sometimes get really different results. And so something that we really want to consider in the immediate future is kind of what equations make sense for the surveys and the fisheries, and also how we want to define those constraints, because it really will change the output of this model. Our model also uses a natural mortality that is constant through time, but studies suggest that natural mortality is likely changing over time, particularly for the Chesapeake Bay region. So the mark recapture model that I mentioned that gave us our occupancy probabilities also estimated natural mortality over time. And what that model found is, while natural mortality was in the Atlantic coast region was fairly constant during their model time frame, which was 1990 to 2020, mortality in the Bay was changing, and that's pretty apparent in this graph here on the left. So this is from Adina Schoenfeld. So the Chesapeake Bay natural mortality in 1990 through 1999 is in the blue here. Chesapeake Bay natural mortality from 2010 to 2020 is here in the orange, and you can see that the recent natural mortality is much higher than the mortality from the earlier years. And what we think a major source of this mortality is from mycobacteriosis, or myco for short. So myco is a disease that's caused by the bacteria Mycobacteria, and myco that affects striped bass was first identified in the Chesapeake Bay in 1997 and causes these skin lesions that you see here on this picture to the right. And since it's been first identified, it's become more prevalent in the Bay, and it's thought to be a major source of mortality in recent years. So something we want to consider going forward is potentially adding in some sort of time-varying natural mortality into the model. And then after our spatial model is complete, we really want to thoroughly test this model with simulation studies. So obviously, before this model can be used for management purposes, we want to understand how this model performs compared to other varieties of spatial models, like a spatially implicit model. We want to know what data is needed for an spatially explicit model of this kind, and also what our limitations are with it. And we also know that the dynamics of striped bass and other species in this region will change and are changing as a result of climate change. And so we want to be able to simulate how our model will perform in the face of these changes. So for example, we may expect the contributions from each of the stocks to change over time, with potentially less fish coming from the Bay and more coming from those northern stocks. And so we'll want to understand how our model might be able to track those changes, if not. And so we still have a lot of testing to do here. And just to kind of wrap up the main ideas of this project and leave you all with just some general conclusions. So sustainable management is often dependent on readily available and reliable stock assessments, and studies have shown how spatial models can have the potential to reduce bias in abundance estimates, because almost all stocks experience some sort of spatial patterning. And most of the data that we actually utilize for stock assessments or population models cover smaller spatial regions, and so they might not be as informative if you try to apply them for the entire range or stock. And so spatial models might be more reasonable to better match the scales that we have data for. And spatial modeling will likely become more important in the future as we see, particularly as we see kind of the increase in the development of wind farms or marine protected areas, and they'll be able to be useful to kind of understand how species are responding in response to some of those new development or kind of changes. And finally, we expect this work will be applied to other ecologically and economically important fish species in the Chesapeake Bay, and be able to improve our understanding of how fish abundance in the Bay has changed over time, and also be used for ecosystem forecasting and other models in this region. And so with that, I just like to give some thanks. First, I'd like to thank NOAA and Sea Grant for funding this project through the PopDive Fellowship. I'd also like to thank the numerous state agencies whose logos are on the screen for providing a lot of data over the last few years. I'd like to acknowledge and thank my lab mates in the Wilberg lab, my advisor, Jenny Neslage, the Secor Lab. I'd also like to acknowledge and thank Rob Latour and Nadina Schoenfeld for their collaboration, particularly with their marker capture model that we use in this model. I'd like to shout out and give thanks to my NIMS mentor, Amy Schuler, for her kind of guidance and mentorship over the years. I'm super excited to be able to continue to work with her in my last year of my fellowship. And I'd also, last but not least, like to acknowledge and recognize Katie Drew with ASMFC and Gary Nelson with the Massachusetts Division of Marine Fisheries. They are the current lead assessment scientists for striped bass, and their guidance and expertise has been super helpful for us as we kind of develop this model, and their collaboration has been super great. So with that, I just like to thank everybody for listening, and I'd be happy to answer any questions if there are any.